Welcome back everyone. Today we learn about blood vessels and blood. We will look at the artery, vein, and capillary. First, the width of the lumen. For the artery it varies, for the vein it is wide to encourage blood flow, and for the capillary it is narrow to permit passage of red blood cells in single fiber. Second, the cross-section. For the artery and capillary it is circular, whereas for the vein it is oval. Third is the diameter of cross-section. For the artery it is between 0.1 and 10 mm, for the vein it is between 0.1 to 20 mm, and for the capillary it is 0.0008 mm. Fourth, let's compare their role. The artery carries pressurized blood away from the heart to other parts of the body. The vein carries low-pressure blood from other parts of the body to the heart. The capillary is responsible for the exchange of nutrients, gases, and waste products. It also connects arteries and veins. Fifth, their physical structure. The artery has thick, elastic, and muscular walls. The veins are thin, less elastic, and has less muscular walls. The capillary is thin, at most having one cell thickness. Sixth, their permeability. Both the arteries and the veins are not permeable, but the capillary is semi-permeable. Seventh, the flow rate and pressure. In the artery, blood flows very fast in spurts and at high pressure. In the veins, blood flows slow, smooth, and at low pressure. In the capillary, blood flow is faster and at higher pressure at arterial end. Eighth is the presence of valve. There are no valves in the artery and capillary, but veins have semilunar valves to prevent flow from reversing its direction. Lastly is the oxygen content. All arteries carry oxygenated blood except the pulmonary artery. All veins carry deoxygenated blood except the pulmonary vein. The capillaries carry oxygenated and deoxygenated blood from the artery to the vein. Now, let's look at the tissue fluid and capillaries. Tissue fluid refers to blood without red blood cells, plasma proteins, and some white blood cells. The cell forming the thin wall of the capillaries don't fit together tightly, hence there are gaps that allow plasma and white blood cells to leak out to coat the tissue and cells. The red blood cells cannot move through the gaps since it cannot alter its shape to slip through, and large plasma proteins cannot pass through the gaps. The function of the tissue fluid is as follows. Firstly, oxygen and digested food materials diffuse from the blood in the capillaries, across the walls of the capillaries, into the tissue fluid, then into the cells. Secondly, waste products diffuse from the cells into the tissue fluid, across the walls of the capillaries, then into the blood in the capillaries. Lastly, it regulates and maintains the temperature and external environment of the cells so that they can perform optimally. We will now study the blood in greater detail. Let's begin with what are the components of the blood. First we have plasma, which takes up 55% of whole blood volume. The plasma transport blood cells, ions, soluble food substances, hormones, carbon dioxide, urea, vitamins, and plasma proteins. Second, we have the white blood cells. They carry out phagocytosis, antibody formation, and tissue rejection. Third, we have the platelets. They cause clotting and soluble fibrinogen to change to insoluble fibrin. Lastly, the red blood cells, which takes up 45% of whole blood volume. They are responsible for oxygen transport as they contain hemoglobin. The blood has two main functions. The first being the transport of materials such as oxygen and carbon dioxide, nutrients, nitrogenous waste, hormone, and heat. The second being for defense or immunity, for instance blood clotting, phagocytosis of white blood cells, and antibody production. Let's explore the components of blood in more detail. The process of blood clotting is as follows. The injury to the lining of blood vessel exposes connective tissue, causing platelets to adhere. A platelet plug is formed in fibrin clot with trapped cells. Do note that platelets themselves are fragments of an originally larger cells, hence they are not cells. The white blood cells or leukocytes can be classified into two groups, the lymphocyte and phagocyte. Lymphocytes have large round nucleus, they release antibodies which bind to bacteria or virus to clump them or destroy them. The phagocytes on the other hand have multi-lobed nucleus and engulfs bacteria by releasing lysozymes to destroy them. Thus, the white blood cells play an important role in our defense and immunity. The red blood cells or erythrocyte have various adaptations. It has a large surface area to volume ratio to increase the rate of diffusion of gases in and out of the cell. It is small and flexible such that it can squeeze through small capillaries. The red blood cells don't contain nucleus, mitochondria, and endoplasmic reticulum to allow more space to hold more hemoglobin. But this also means it has a short lifespan for about 4 months. Lastly, red blood cells contain hemoglobin to transport oxygen, and it gives the cells its red color. Let's move on to the blood groups, antigens and antibodies. Antigens are molecules that can cause antibodies to be generated by the body. Antibodies are molecules that bind to antigens. The ABO blood grouping system is based on the presence or non-presence of 2 antigens on the surface of red blood cells in our body. Antigens that are naturally present in the body are called self-antigens. These are tolerated by the body and no antibodies are produced in response to them. The body produces antibodies in response to antigens foreign to the body. In this table we see the 4 blood group and the corresponding antibodies found in their plasma and antigens present on their red blood cells. We will end of this video by looking at blood donation and agglutination. Blood type O is the universal donor, as it is always safe to accept blood from them since their red blood cells have neither antigen A or B. Thus, the recipient antibody will not produce any antibodies in response to blood type O blood donation. Blood type AB, on the other hand, is the universal recipient, as it is always able to receive any blood type since their plasma doesn't have antibody A or B. For example, if recipient has blood type A, it means the recipient has antigen A on their cell membrane. Thus, the recipient will tolerate antigen A and blood will not contain or produce antibody A. However, the recipient produces antibody B and thus will not be able to receive blood type B and blood type AB transfusion. Should the recipient receive blood type B or AB from the donor, it will result in clumping or agglutination of red blood cells. So what does this mean? Well, during the process of blood donation, the donated blood can be separated into its components. The antibodies are contained in blood plasma, which is separated from the blood cells. Therefore, a blood transfusion recipient receives a blood donor's red blood cell only without the donor's antibodies. Meaning that we should only consider the effect of the donated red blood cells interacting with the recipient antibodies found in the recipient plasma. Thus, the overall consideration is the donor's antigen on the red blood cell and the recipient's antibodies. That's all for today. Thank you for the effort to learn with me. Stay tuned for more interesting lessons. See you next time. Bye-bye.